Good morning. My name is Lin Lin. I'm an associate professor in Aarhus University, Clinical Diabetes Center, Aarhus. Today, I'm going to introduce my work on exploring the mammalian brain. I will turn off my video now so we could have a better view of the slides. Our brain is one of the most amazing and beautiful organs in our body, but very few of them were known about it. A lot of diseases are related to brain, and different animal models were generated to explore the mechanism behind it and the potential therapy method. But what would be the most ideal species for animal models for human brain? Monkey, pig, mouse, or rat? There are different aspects that we need to think about. From a genetic point of view, they are all very close to human beings. However, when we look at the size and the morphology of the brain, mouse and rat are too tiny. From the ethical point of view, it is too critical to use monkey. Besides, it takes too long time for breeding and also it's quite expensive. So, in the end, pig win the game. But how much do we know about our brain and pig brain? The answer is, the current understanding of regional specific gene expression in mammalian brain, typically in pig, are rarely explored. To expand the basic understanding of neurobiology, several brain mapping projects were done on basic organization and regional or cellular gene expression of mammalian brain, including mouse and human. For complement the available datasets, in collaboration with scientists from Karolinska Institute and BGI, we have performed a comprehensive, in-depth molecular dissections of various brain regions using multiple transcriptomics method and antibody-based mapping in human, mouse, and pig. This combined approach allows us to identify the regional expression profile of protein codon genes, their similarities and difference within or between different regions or species. The first part of the data were analyzed from the open access database, including genotype tissue expression portal and the functional annotation of mammalian genome 5 and adult mouse brain. These figures summarize what we have included for brain subregions. So 23 subregions in 12 main regions in human, 28 subregions in 12 main regions in pig, and 17 subregions in 11 main regions in mouse. Together with the database, the data were normalized and combined into 5 separate datasets. There are more than 17,000 common genes within the 3 human dataset. In mouse, there are 15,160 genes with 1-to-1 orthologs to human gene, while in pig, there are a little bit less, 14,656 genes with 1-to-1 orthologs to human gene. This is probably due to the annotation of pig genome is not as good as human and mouse genome. When we look at the common genes between the 4 datasets, including human, mouse, and pig, there are around 13,000 genes that we could make the comparison. The big dataset allows us to do quite a lot of comparisons, either between different regions or subregions in the same species, or the same region or subregions from different species. Here you could find the difference of gene expression pattern with the same species. Expression data from the various brain regions of the 3 species were summarized into 10 main regions. This UMAP plot helps us to see the distance of expression pattern from any 2 samples by deducting the multiple dimension of variable genes into 2D plot. We could see the samples from the same region were clustered together, and the cluster with more similarities were plugged closer than the others. For example, Cerebellum shows quite a distance to the other subregions. The hierarchical cluster tree shows a similar structure, with the cerebellum as outlier in all three species. Of course, this is not all what we could see from the database. We could compare any of the genes expression in the dataset within or cross species, like here, the first row in figure A. And we're not satisfied about the giant dataset. The RNA-seq expression data provide a sensitive quantification method for transcriptome information. Although these data have the disadvantages, which is it's a mixture of the cell type they are analyzed. The in situ hybridization data provide spatial expression data on the single cell level. But this probe-based method is less quantitative than the count-based RNA sequencing method. In addition, for selected protein, an immunofluorescence protein distribution map was generated, allowing visualization of the distribution of protein, the final product from the centroderma with functions on a cellular level, including neural process with high spatial resolution. Another advantage of this protein staining is that anatomically stacked imaging could be generated, and this has allowed us to annotate more than 120 regions and subfields of the brain. We could also generate the 3D imaging based on the gene expression. Together, the three complementary datasets, including RNA-seq, in situ hybridization, and immunofluorescence staining, provide us genome-wide regional profile of the protein coding genes and their expression in different regions and subregions of the brain. In order to share our dataset, we launched a Brain Atlas database to present and integrate all the data reported here. And this is an extension of the HPA Human Protein Atlas portal, with a brain-centric summary page for each gene with expression data in human, pig, and mouse brain regions. The resource is presented with an expression summary for all protein coding genes. For selected genes, the distribution of corresponding protein is visualized by antibody-based protein detection. On the top of the slides, you could find the link of the page. In the Brain Atlas project, for RNA detection, we have used two different approaches. One is in situ hybridization. It allows us to look at both the location of RNA and also it is at the single cell level. But unfortunately, not in a high throughput manner. Another method that we use is RNA sequencing for high throughput detection of genome-wide gene expression. However, the RNA was extracted from a mixture of cells, so the expression level we see is the average of them all. It might be okay for the cell line, but in vivo, even in a tiny piece of samples, there are quite a few different cell types. It is like in this figure. In the tube, there are quite a lot of balls with different colors. But if we blend the balls, we will lose the original beautiful color and can only see gray. How about only use one cell to generate one library? Now a lot of method has been developed for RNA sequencing and single cell resolution. For all of the methods, the key point is to capture the mRNA individually and label different cells with different barcode. So we could do the in silico selection to isolate the sequencing result from the single cell. To achieve this purpose, people tried different solutions. You can see in the beginning, Single cells were manually picked up and used for mRNA capture and library generation. Think about if you're working with a few hundred of cells. It's quite a lot of work already. And also it's very expensive for all the reagent. And later, different method has been developed, which is to provide the physical barriers between cells. So the reaction could be done separately. The barrier could be different things. It could be the wall of the wells, the microfluid channels, or the oil drops. After the mRNA from the same cell were labeled with the same barcode, the cDNA will be pulled together and used for library generation. After sequencing, the sequence will be aligned to reference genome, counting the gene expression and split by cell. The most popular product in the market now is the 10x genomics, which was based on the oil drop system, could allow us to generate one library with more than 10,000 of cells and split the cell afterwards. One good application I would like to work with is, of course, to see the single cell profile in pig, especially in brain and muscle. This project is undergoing process, and we're still analyzing the data. So today I would like to share another project we have worked with, single-cell RNA sequencing on endothelial cells. Vascular is one of the largest organs in our body, and it existed in almost every tissue. And we know the endothelial cells are with high heterogeneity. Within the tissue, there are different types of endothelial cells. There are artery, and there are capillary and veins. And for different tissues, and they show different morphologies. For example, the junction between endothelial cells were much tighter in brain than liver, as you can see from the images. In collaboration with Ioana Kathika from VAB, who recently joined Aarhus University, we have done the mouse endothelial cells atlas project together. In briefly, the endothelial cells were isolated and max enriched and FACS purified from different tissues in mouse and used for single-cell RNA sequencing with 10x genomics technology. In total, we have get the sequencing data from 32,567 endothelial cells from 11 tissues, which including 1,745 lymphatic endothelial cells and 30,822 blood vascular endothelial cells. Worth to note, in this UMAP, we have seen in Brain Atlas, one dot is one sample, but here what we see in the t-SNE plot, one dot is one individual cell. The endothelial cells from the same tissue were clustered nicely together. Also, different markers were well identified for different tissues. When we performed the correlation analysis, we could see the endothelial cells from testes and brains are quite like each other, which is identical to the morphologies. The comparison could be between different tissues, which we could do as well with the bulk RNA sequencing. The beauty of single-cell sequencing is that we could explore the heterogeneity within the tissue and see how many subtypes of cells that we could see from one pool of samples. Well, due to the limited time, today I'm not going to go through all more projects that we have worked with single-cell RNA sequencing. So if you have any questions, you're welcome to contact me by email, which is in the first slides, or ask in later discussion. Also, I have one question or homework to you. Now we could look at the gene expression at different resolutions, the pool of the cells and single cell, and also at different scales, single cell, tag genes, or genome-wide. Since now we have this fancy technology which allow us to do the genome-wide RNA expression profiling at single cell level, could we add one more information or one more dimension, which is the location of the RNA, so we could get all the information in one run? Let's discuss more details in the discussion set. Bye, see you.